So I welcome you to the third tutorial session of this course, Introduction to Cell Biology. And today we will try to kind of revise and discuss the topics that were taught in this third week of the course. And so to get started, again, I will just run you through the summary of what has been taught in this third week. So initially, in the previous week, we had, in the previous 2 weeks, to be honest, we have discussed a detailed, uh, we have taken a detailed view of different aspects of central dogma and molecular biology of DNA replication, transcription, translation, as well as after the linear polypeptide chain consisting of amino acids where the amino acid residues are linked together via covalent peptide bonds, then how the linear polypeptide chains, which are being which are being translated as a result of reading of the mRNA by the translation machineries, that is the ribosomes, how do they fold? What are the principles, what are the driving forces that govern the protein folding? And how does how do these linear polypeptide chains fold into their three-dimensional structures, which is necessary for their biological functions? So one of the very important classes of the proteins, which are the effector molecules of the cell, which are the workhorses of the cells, are actually enzymes, right? However, enzymes can be both proteins as well as there can be RNA enzymes, or which are called the ribozymes. But most of the enzymes have a large category, large part of these enzymes are protein molecules, which are actually biocatalysts. These are the catalysts of the cell, which catalyze a particular chemical reaction within the cell. So what they essentially do is they influence the kinetics of the chemical reaction. They make the chemical reaction happen at much faster rates. They do not influence the equilibrium of the chemical reaction. That is, they do not influence the extent to which the chemical reaction proceeds, but they influence the kinetics, the rate at which the chemical reaction proceeds, right? And that also with a very high specificity. That is, the enzymes bind a certain set of substrates and then catalyze the formation of a certain product, right? Now, as you can imagine, there's these enzymes, right? These enzymes are proteins. So basically, um, the linear polypeptide chains consisting of amino acid residues linked by these peptide bonds, which eventually fold into certain three-dimensional structures. And these structures also are necessary for ensuring this specificity, that only a few amino acid residues which are present in this active catalytic site are able to recognize the substrates and also then catalyze the chemical reaction. So actually, if you see a chemical reaction, then there is always if you look at the free energy diagram of a chemical reaction, then you will find that the products have much lesser energy than the substrates, but there is always there is a free energy barrier, that a hump that exists that prevents like you need to provide energy to overcome that activation energy barrier so that the substrates can get converted into the products. So enzymes lower the activation energy barrier and therefore they catalyze the formation of products from substrates. Now there is a model which is called the induced fit model. That is, since that the protein the proteins are not fixed molecules. They're not like statues. They're continuously fluctuating to a certain extent. So whenever there is a ligand that is coming in, which will be the substrates in this case, so the ligand also actually changes the protein conformation to a certain extent. And so when the ligand comes, the enzyme also changes accordingly, and there is a binding. But there is a specific binding because only there is a set of amino acid residues that are present in this active catalytic site, which are able to drive those interactions that are necessary for binding the substrate. And then these active site amino acid residues, they will have those R groups that has those functional groups. Like it can be the OH groups, the SH groups, right? Or different other groups, like it can be positively charged residue or a negatively charged residue, which can turn out to be very important for binding metal ions, which can be important cofactors for driving some important reactions. So then they catalyze basically the reaction of these substrates by lowering the activation energy and thereby increasing the kinetics of your chemical reaction. Okay. Now, so there was a very brief discussion on enzymes, and then there was like you were given an overview of two other like you had already learned about the nucleic acids, that is DNA, RNA, and you had learned about the proteins. So only two other major biomacromolecules were left, that were lipids and carbohydrates. So lipids are um, lipids can actually be categorized in several can be categorized to several different categories. For example, lipids can be glycerolipids or glycerides. It can be glycerophospholipids. It can be sphingolipids. It can be cholesterol and various other. So lipids can be categorized into several different kinds, right? Primarily, if you look into like the most abundant lipids, which are the glycerolipids or glycerides and the glycerophospholipids, then you will find that uh, it consists of a hydrophilic, like in the entire lipid molecule. We will go into the details, like into the chemical nature of these lipid molecules. So it consists of a hydrophilic head group and the long hydrocarbon chains, which are the fatty acid chains, which are hydrophobic, right? So hydrophilic means that it can interact with water via hydrogen bonding. Therefore, it loves water. That is, it's called hydrophilic. That means it will have those hydroxyl groups which will be able to interact with water via hydrogen bonding, and therefore is hydrophilic, one part, whereas the long hydrocarbon chains of the fatty acid residues, which consists of long carbon chains. So they don't have the hydroxyl groups and they are not able to interact with water via hydrogen bonding. So they actually hate water and they always try to assemble in that manner that the hydrophobic residues are always hidden from the water molecule. So that's why what happens is, for example, if you take this lipid molecule and if you for example mix it in water, and gradually if you increase the concentration of these lipids in water above a certain critical threshold concentration, so gradually what happens is that the water molecules which are capable of interacting with this hydrophilic head group, they kind of are in a very dynamic state where the water molecules are hydrogen bonding and like new water molecules are coming in, old water molecules are going off. Like there is a very dynamic exchange of water molecules because of the interaction between the water molecules and the head group. But for the hydrophobic chain, since it is not able to interact with water molecule, what happens is a certain set of water molecules, they form one cage around the hydrophobic chain. But there what happens is a certain number of water molecules, their entropy is lost because they are not now free to randomly move. And these water molecules that make the cage around this hydrophobic tail, which is basically the fatty acid chain, so that causes a net lowering of the entropy of the entire system. But that is not favorable because, as you all know, that according to the laws of thermodynamics, the system always tries to achieve the max entropy. So because of that, what happens is when you gradually keep on adding these lipids, so what happens is more and more number of water molecules form this cage around this hydrophobic chain, and their entropy is reduced. So the net entropy of the system keeps on reducing even further. And so there what happens is that these lipid molecules self-assemble. They are not able to interact with water, but the hydrophobic chains can interact with these hydrophobic chains, right? There can be hydrophobic interactions and interactions that are preventing the hydrophobic chains from getting exposed to water. So they assemble themselves in the form of these lipid bilayers, right? They actually assemble themselves in the form of a lipid bilayer, where you can see that they are assembled in such a manner that only the hydrophilic head groups, right? Only the hydrophilic head groups, is red head groups, they are getting now exposed to water, which is perfectly fine because they are hydrophilic. They can interact with the water by hydrogen bonding. And this hydrophobic fatty acid residues, they're actually tucked inside the lipid bilayer. So there is hydrophobic interaction between the fatty acid chains, but they are hiding from water, so that the water entropy is not dropped and the system entropy is maximized. Okay. So now let's look into the chemical nature of okay. The chemical nature of the lipids, right? So, for example, in these glycerolipids, which is a glycerolipids and glycerophospholipids, both. So primarily what happens is there's a glycerol molecule, okay? And then there are the fatty acid chains. So fatty acid, so that's why you can see that it will always be on one end, it will be COOH, right? And then there is this long hydrocarbon chain. You can see carbon, 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 carbon. There is a long chain of carbons, long hydrocarbon chain of hydrocarbons, long hydrocarbon chain basically with one COH at one end. So this is your typical fatty acid. Now the number of number of carbons in this hydrocarbon chain will be different. That leads to different fatty acids. It can be oleic acid, it can be myristoic acid, it can be palmitoic acid, etc. So now what happens is between one glycerol molecule and fatty acid molecule, between one hydroxyl group, right? Between one hydroxyl group of this glycerol and one hydrogen of this fatty acid, this actually goes out in the form of water, and one chemical bond, right? One chemical bond is formed between the glycerol and the fatty acid. A covalent bond is formed. So if you look at this covalent bond, so it will be C double bond O and the fatty acid. So it will be R C double bond O, single bond O R. That is the ester bond, right? So now one ester bond is formed between this glycerol and the COH of fatty acid, which is a hydroxyl group and COH basically of the fatty acid to form an ester bond. So that links the glycerol molecule to the fatty acid. Now this glycerol, since it has this hydroxyl group, it will be the glycerol forms a hydrophilic head that we were mentioning there. And this fatty acid, since if you see, it has no hydroxyl group, right? It has only these carbons with which the hydrogens are attached. So they form the hydrophobic fatty acid tail, which is not capable of interacting with water via hydrogen bonding, right? Now, in case of phospholipids, what happens is, like, so in glycerolipids, you can have two fatty acids attached in these two positions. In case of glycerophospholipids, what happens is fatty acids are attached to these two carbons of the glycerol via these ester bonds, and to the third carbon is attached one phosphate group, right? Is attached one phosphate group.
Sir,
Hmm.
sir,
Yeah, so both.
yes, sir. Sir, can I ask one question from previous topic?
You can ask.
Sir, sir, in the replication, DNA replication,
Hmm.
the DNA template, first the RNA primer goes and joins, starts the joining process, means the replication process.
Hmm.
Why the
Hmm.
RNA is going? And first it will bind with A, then it will bind to T only. Why RNA is doing the first joining process?
That is because of the enzyme actually. So when this primase comes, it's this enzyme primase which after the helicase opens the two strands of DNA, and the primase actually polymerizes a very short stretch of these oligonucleotides to create the single strand double strand hybrid junction. To create the single strand double strand hybrid junction, because the DNA polymerase recognize only this double strand single strand hybrid junction. It needs a short oligonucleotide template having this 3' hydroxyl group which it can extend. So that's why in initiation this is needed. But after initiation, what happens is the DNA polymerase 1 replaces these nucleotides with deoxyribonucleotide residues, complementary deoxyribonucleotide residues, and finally the gap is also joined. I mean, here there will no gap, but in case of the Okazaki fragments, the gaps are also ligase. They are joined by the ligase enzyme. So in short, the answer is that primase forms a short oligonucleotide of ribonucleoside triphosphates to initiate the DNA replication, because DNA polymerase is able to recognize only the single strand double strand junction.
Okay, sir, understood.
You understood? The single strand double strand junction is necessary. Otherwise, if this oligonucleotide is not there, then you just have the single DNA strand, which the DNA polymerase is not able to recognize. And not only it's a question of recognition, but also it has no short nucleotide with this 3' OH group, which it can elongate. I mean, to which it can add the new DNTPs, because this 3' OH group is necessary for adding new DNTPs. Because 3' OH is the only nucleophile that will attack the 5' phosphate of the incoming DNTP to form the phosphodiester bond. Understand?
Yes, sir, understood.
Okay. So in case of this glycerophospholipids that I was mentioning, there what happens is that in case of glycerophospholipids, you have two fatty acid chains. Okay, you have two fatty acid chains which are joined to these two carbons. Basically, these two carbons of the glycerol, because of formation of these ester bonds between the fatty acid and the glycerol. And the third carbon, what happens is one ester bond is formed between the phosphate and the glycerol moiety. And to the phosphate, on the other hand, with one OH group of the phosphate moiety is added some other residues, like it can be ethanolamine, it can be serine, and that gives you the glycerophospholipids, where you have two fatty acid residues, a glycerol molecule, a phosphate chain, and then another compound. It can be serine, it can be ethanolamine. That gives you different phospholipids. Okay. Now, this in case of glycerophospholipids, so this glycerol as well as the phosphate actually forms the glycerol phosphate. And for example, if you have this serine or ethanolamine, then this forms your hydrophilic head group, whereas this fatty acid residues, they actually form your hydrophobic tails. Okay. Yeah, so this is actually one representation. For example, even in glycerophospholipids, as I already mentioned, that you can have several different types of glycerolipids or glycerides, where, like, for example, monoglyceride, diglyceride, and triglyceride. But what they essentially differ in is how many fatty acid residues are attached to the glycerol moiety. If a single fatty acid residue is attached, it's a monoglyceride. Attached by this, you can see this ester bond, this covalent ester bond. If two fatty acid residues are attached to two different carbon atoms of the glycerol moiety via formation of these ester bonds, then they are diglycerides. And if three fatty acid residues are attached to three different carbon atoms of the glycerol molecule, then it's a triglyceride. Now, depending on the number of carbon atoms in the hydrocarbon chain, they can be of three categories: short chain fatty acids, medium chain fatty acids, and long chain fatty acids, right? So short chains having 2 to 5 carbon atoms, medium chains having 6 to 12 carbon atoms, and long chains having more than 13 or more than 13 carbon atoms in the hydrocarbon chain. There is one now among these fatty acid residues, among this fatty acid chain, uh, the fatty acid molecules can be again classified into two different categories. Like they can be saturated fatty acid residues, or they can be unsaturated fatty acid residues. So this division or classification is dependent on this fact that whether in this hydrocarbon chain, all the carbon atoms that are present, they are linked by a single, all of them are linked by a single covalent bond, or between some carbon atoms, whether do you have a double bond or not. So if all the carbon atoms of the hydrocarbon chain of this fatty acid are linked by single bond, then they are called the saturated fatty acid. Okay. Now, as you can think that since it's saturated fatty acids, they can stack very nicely with one another via hydrophobic interactions. Okay. Because of that, they pack very well. Since they stack very well, they make the membrane much less fluidic, right? That is, if you gradually lower the temperature, okay, the saturated, I mean, the saturated fatty acid chains, they can pack very nicely, and they make almost a solid. If solid, that means not really solid, very less fluidic membrane. Yes, very less fluidic membrane, where there is not much of this three-dimensional mobility of these lipids within the this membrane, within each leaflet of the membrane. Whereas in case of unsaturated fatty acids, what happens is, so unsaturated fatty acid essentially means it contains at least one or more than one double bonds among the carbon atoms of the fatty acid hydrocarbon chains. Okay. So if it contains a single double bond, then it's called monounsaturated fatty acid. If it contains two or more double bonds, then it calls polyunsaturated fatty acids. But what happens because of introduction of these double bonds is that you can see that the saturated fatty acid chains were absolutely linear, because of which they can pack very nicely. However, but these unsaturated fatty acids, they, because of the presence of this double bond, there's a kink in their structure, right? A single double bond, there is this kink. If it's a double, like if there are more than one, that is two or more double bonds, then if it's more kinked. Since it's more kinked, it cannot pack very nicely, right? Because they are already kinked. If they are linear, they can pack very nicely. However, if they're, since they're kinked, these unsaturated fatty acid residues, they cannot pack very closely. Because of that, even upon lowering of the temperature, they they keep the membrane much more fluidic, right? So this balance of saturated and unsaturated lipids within the membrane is very essential for maintaining the maintaining various physicochemical properties of the membrane, especially membrane fluidity, because which in turn will influence, for example, the mobility of the proteins in the lipid membrane bilayer, as well as the protein conformational changes that are necessary for protein functions. As well as, on the contrary, if you think from the perspective of membrane integrity, membrane stability, then these saturated fatty acid residues are very necessary for the membrane stability and integrity. For example, upon increasing temperatures, the saturated you need the saturated fatty acid molecules to make the membrane exist, because they can pack very nicely. So you need very high temperature for disintegrating the membrane. Whereas if it was just the unsaturated fatty acid residues, then at a moderate temperature increase, membrane integrity would have been lost. So a balance of these saturated and unsaturated fatty acid residues or molecules in the lipid membrane bilayer is very necessary for having this balance of membrane integrity as well as membrane fluidity, which is necessary for protein function and various membrane functions. Selective permeability, basically. Okay. Now the last type of bimacromolecule that was discussed in this week are the carbohydrates, right? So carbohydrates or sugars, so they consist of this like there are aldose sugars. So in case of carbohydrates, the most simple sugars, the units of carbohydrates are monosaccharides, right? The monosaccharides can consist of different number of carbon atoms. It can be consisting of 5 carbon atoms. Then they are called the pentose, which we have already seen. That examples of pentose are ribose and 2-deoxyribose. They can consist of 6 carbon atoms. Then they are called the hexoses. which are your most common monosaccharides like glucose, fructose, etc. Now, among these carbohydrates, these monosaccharides basically, again, they consist of an aldehyde group or a ketone group. Okay, the ones that consist of aldehydes are called aldoses. The one that consists of ketone group are called ketoses. That's also classification. And after that, all the other carbon atoms have hydroxyl groups attached to them. Okay, so now these monosaccharides, they actually the two monosaccharide molecules can link together via formation of a covalent bond, which are called the glycosidic bonds, right? Which are basically ether bonds between two. They're actually called these glycosidic bonds because of removal of one water molecule between this COH and another COH. Okay. So if you see that, what I was trying to mention is this, that the most simple sugars are the monosaccharides, which consist of a single sugar molecule, can have different number of carbon atoms, can be aldoses or can be ketoses. These monosaccharides link together via formation of this glycosidic bond, right, to form these disaccharides. Now, there can also be oligosaccharides, where there can be several disaccharides which are linked by glycosidic bonds, or there can be these basically monosaccharides linked by glycosidic bonds to form chains of higher order carbohydrates having 3 to 9 monosaccharide units, smallest units basically. Then there are called oligosaccharides. If they have 10 or more monomeric units linked together by glycosidic bonds, and they're called polysaccharides. For example, starch. So monosaccharides, the most common examples are glucose, fructose, galactose. Right. So while glucose and galactose are actually aldoses, fructose is a ketose. You will see that their prevalence in different food items. For example, galactose is a milk sugar. Fructose is very commonly found in fruits, honey, etc. Whereas glucose is the most important sugar for us and the major source of energy, especially for the brain. And they cross this blood-brain barrier for reaching the brain, which uses glucose as a major source of energy. All our cells, to be honest, uses glucose as the main source of energy. But brain has, in our case, the brain has a specifically high demand of glucose as the energy source. Now, in case of disaccharides, for example, these different monosaccharide residues, you can see, for example, two glucose molecules can get linked via formation of a certain type of a glycosidic bond to form maltose, whereas glucose and galactose are joined together via glycosidic bond to form lactose. And the sucrose, which is our table sugar, the sugar that we most widely use in our tea, coffee, and various other preparations, so that is basically glucose and fructose linked together by a particular glycosidic bond, right? These are your disaccharides. So
Sir,
this was Ashwam.
sir, means all non-saccharides will have double bonds. Non-saturated, sorry, non-saturated will have double bonds.
All unsaturated will have double bonds. Yes, it can have a single double bond, or it can have more than one double bonds. That is two or more double bonds. If it has a single double bond, then it will be called monounsaturated. If it has more than one, that is two or more double bonds, then it's called polyunsaturated.
Means it is compulsory to have
No, it
double
is
bond.
not like that. The thing is that if you look at all the naturally occurring fatty acid molecules, then you will find that some of these fatty acid molecules are saturated. That is, they don't have any double bond. Okay. And some of these fatty acids have double bonds, so they're unsaturated. Now, what you are asking, maybe I am understanding your question. So if you look at the membrane composition, membrane lipid composition, right? They have glycerolipids, they have glycerophospholipids, sphingolipids, cholesterol, many things. But among these glycerophospholipids, definitely there will be a balance of saturated and unsaturated fatty acids. So there I will say yes, it is necessary to have unsaturated fatty acids as well. Otherwise, your membrane fluidity cannot be maintained. So the
Okay, sir,
membrane
understood.
has both saturated and unsaturated fatty acids in the glycerolipids, in the glycerophospholipids that make the membrane.
Okay, sir, understood.
Okay. Okay. So after covering all the different important biomacromolecules, in this third week, there was a jump actually. So the next topic that was being discussed is there was an introduction to genetics, to Mendelian genetics and non-Mendelian genetics, right? So many of you, I think, that have read Mendel's work in higher secondary and in several in different stages of classes to certain extent or other. So Mendel did some fabulous work to understand the laws or rules of inheritance of traits or phenotype by working or observing the uh, patterns of inheritance of traits or phenotypes in the garden pea plant. Now, when Mendel did the work, did his experiments basically, he, I mean, he didn't knew, and not only he, but the community didn't knew anything about the concept of genes or genotype. That is, nobody knew that DNA is a genetic material. Nobody knew that a section of the DNA, a fragment of the DNA having a certain nucleotide sequence, acts as the gene which can encode for a gene product, which can be one RNA molecule or one protein molecule that eventually helps the cell achieving a certain function. And like any mutation in the gene leads to different leads to altered generation of this gene product. It leads to basically alteration of the gene product, which leads to alteration of the phenotype. He didn't know any of these things. He just could see the phenotypes. He could just see the manifestation, the kind of physical manifestations of some parameters, which definitely had their origin in the genotype or the genetic composition of the organisms. But he just observed that, for example, from very simple perspective, that a pea plant gives rise to a pea plant. Now he just went a bit more deeper that if whether a tall pea plant is giving rise to tall pea plant, whether a pea plant having yellow pods are giving rise to a pea plant that also gives a yellow pod, like that. So he was observing phenotypes, and he was observing how these traits or phenotypes were getting passed on from one generation of the organisms to the next generation of organisms, and therefore came up with a set of rules, which he thought are the rules that govern heredity or inheritance of traits or phenotypes. He, however, mentioned that there should be something underlying these phenotypes, and he used to call them factors. So, essentially, Mendel studied various different phenotypes of the garden pea plant, right? For example, these are the phenotypes. That what was the seed coat color? What was the seed coat color? Um, seed color, for example, yellow or green. What is the seed shape? Smooth or wrinkled? What is the pod color? Green or yellow? What is the pod shape, right? Or, for example, what is the height of the plant? Stem height, right? Tall and short. And what is the flowering position, right? Axial, terminal. So basically the phenotypes that he studied are these: the seed color, seed shape, right? These are your phenotypes that he studied. But while he tried to observe, for example, stem height, right? He has so many different pea plants within in his garden, and there is automatically this diversity among the genes that is existing, which he realized that there is a diversity of, for example, the phenotype is seed color, but there is a diversity in this fact that some of the seeds are yellow and some of the seeds are green, right? So the phenotype is same height, but some of the plants are tall, some of the plants are short. In case of pod color, for example, he realizes some of the plants have green pods, whereas some of the plants are having yellow pods. So essentially, what we know today is that in diploid organisms, right? In diploid organisms, you have two sets of each gene which are present in a pair of chromosomes. So for each chromosome, you have two copies, right? And at each locus of the chromosome, right? At each locus of the chromosome, you have a particular, like you have the gene at a particular locus of a pair of homologous chromosomes. Now, in these two chromosomes, you can have different variants of the gene. So there are different variants of the gene, right? So the gene is basically a fragment of DNA having a certain nucleotide sequence which encodes for a gene product. But there are always mutations that can crop in, right? And because of that, there are different variants of the gene. So these different variants of the gene which are present at the same position, right? At the same position or at the same locus of these homologous chromosomes. So they are called the alleles. Okay. And basically, if you really want to realize that what are the variants of one particular gene, that is, what are the alleles that are present, then you will not be able to appreciate this if you just see one organism or one individual or one pea plant. You have to look into all the pea plants of your garden. Then only you get to realize that, okay, if the frequency may be different. Maybe if you see 100 pea plants, you may be seeing that, okay, 90 pea plants are having green pods. But you will see that there is also one more possibility or one more variant, um, where the plant is actually having yellow uh, seeds, basically yellow peas, right? So that helps even understand, that actually kind of indicates that this gene may have different variants or different alleles, right? Now, how do these different alleles eventually lead to these different phenotypes, that we will try to understand in one of the very classic Mendel's experiment, which is called the monohybrid cross. So as per our current understanding, what we understand is that if you remember the cell cycle, then in mitosis, as well as in case of mitosis or equational division, or in meiosis, which is even more important to understand in this case, because meiosis is the type of cell division which is involved in giving rise in generating your gametes, right? Which are the reproductive units which are necessary for inheritance, um, for transferring the genetic information from one organism to the next organism, which is the offspring. So in meiosis, what happens is, first there is replication. So initially diploid organism, you have two copies of a particular chromosome. For example, if you take the chromosome number 21, then there are two copies of chromosome number 21 in each of your cells, right? Now, for meiosis, what will happen is, first there will be replication. So in each of these chromosomes, there will be two sister chromatids, right? There will be two sister chromatids, which are formed as a result of replication of the initial parent chromatid. And then what will happen is, there will be segregation, right? There will be a segregation of each of these like chromosomes to each gamete. There will be four gametes, each will have one one chromosome. And finally, so your gametes will be haploid, right? But eventually, when the male gamete and the female gamete will fuse, there will be fertilization. There what will happen? Again, the diploid organism will be formed, where one of the homologous chromosomes will come from the father, and the other homologous chromosome will come from the mother. Right? One paternal chromosome, one maternal chromosome. One paternal copy of, for example, chromosome number 21, and one maternal copy of chromosome number 21 to give rise to the diploid, again, homologous pair of homologous chromosomes 20 of chromosome number 21. So now, if we just try to see what Mendel said, what was Mendel's experimental setup? So pea plant is actually a hermaphrodite. What that essentially means is that it is capable of doing self-fertilization, because it has both, its flowers have both the male reproductive organ as well as the female reproductive organ. So the pollen from, so each flower is capable of fertilizing itself. The pollens from the anther can come to the stigma of the same flower, and there can be self-fertilization. So if there is self-fertilization, then it's actually it's a continuous self-cross, right? And that will not tell you the pattern of inheritance. So Mendel actually did a cross-fertilization experiment. He categorically maintained this cross-fertilization experiment, where he will cut away the anthers just after, at the very early stages of flowering, so that there is no self-fertilization. Instead, what he did is he collected these anthers from one flower and transferred the pollens from the anthers of one flower to the stigma of another flower, like to make the cross-fertilization between two plants that he wanted to cross. Okay, this you should keep in mind while understanding Mendel's experiments, because which is important, because P is a hermaphrodite. Like, it has both the like even in each of its flowers, it has both the male and female reproductive organs. It's capable of self-fertilization, but Mendel always ensured cross-fertilization. What Mendel postulated from his experiments were that behind the phenotypes, there must be some underlying factors which he couldn't understand, he could not reveal or um, unravel the chemical nature of the factor, but he could understand, he could predict, he could um, observe, and he postulated that the factors exist in pairs. That is, the pea plant was a diploid organism that exists, and here the factors existed in pairs. And these factors do not mix or contaminate. That is, they have individual identities, and these individual identities are always preserved. They never mix with one another or contaminate with one another. Their individual identities are always individual. They're always preserved. Sometimes they get to kind of, express themselves and express in terms of regulating the phenotype. Sometimes they don't get to, for some reason or other, which we'll see why. But they're always, if they're there, they never mix or contaminate with one another. They always maintain their individual identities. And after, if sometimes they, like, in some cases they might not be able to express themselves, if you may not realize that they are there from the phenotype, but upon the suitable condition arising, they can again express themselves in and regulate the phenotype. So they never mix or contaminate each other, and they're always segregated. And he could also kind of postulate that there must be different variants of these factors, right? The factors which we currently call genes. So he could predict that there must be variants of these genes which are called the alleles, right? Now, this concept, he also postulated this concept of dominance and recessiveness. What that really means is that one particular gene, like for example, so the organism is diploid, right? That means it has a pair of homologous chromosomes. That is two chromosomes, right? For each particular chromosome, it has two copies. That means at a certain position of each chromosome, that is at each locus of each chromosome, it will have two copies of a particular gene. Now, these two copies can be identical, or these two copies may not be identical. So if they are not identical, that is there are two at the one particular locus, there are two different alleles, that is two different variants of the single gene. Then he found out in his experiments that if the two gene variants or alleles present in the same locus of homologous chromosome are not identical, then only one of them is able to express in terms of the phenotype and the other is not. So the one that is able to express itself in terms of the phenotype is called the dominant, and the other one is being called the recessive. Okay, so it's basically relative terminology. But again, always remember that they are not mixing or they are not contaminating each other. They have individual identities. Upon further crosses, it may happen that if the recessive genes are again becoming homozygous, that is, if this situation arises, that in these two copies of homologous chromosome, if both the genes that are present are identical and recessive, then again this recessive trait or recessive phenotype can be re-expressed. So that is, they are not losing their individual identities, they are not mixing. Now, this law of segregation. So, as I have already mentioned several times, that these alleles are not mixing or contaminating, and they segregate randomly into the gametes, which are the haploid, right? Because of this meiotic cell division, which I said in this slide, that in meiotic cell division, what will happen is basically there will be 4 gametes, right? And these sister chromatids, like these sister chromatids of the homologous chromosomes, basically they will get separated and segregated into the haploid gametes randomly, right? There's no bias. That is the law of segregation, and these alleles or genes, they are never contaminating or mixing with one another. So that is the law of segregation. Now is the fourth observation from his experiments was of independent assortment. So in independent assortment, basically what he meant to say is that that, for example, two different phenotypes. You have two different phenotypes. For example, you have this seed color, right? Which is, you have one trait or phenotype which is a seed color, and you have other trait or phenotype which is the same height. So he found that same height inheritance is independent of seed color inheritance. So how the seed color inheritance is happening is not influencing the stem height inheritance pattern. That means the stem height inheritance pattern and seed color inheritance pattern are independent of one another. So that is possible. So now how is that possible? So regarding that, he mentioned that the genes which are, or the factors basically, which according to Mendel are probably regulating this stem height. The factors or the alleles present in a particular locus of a certain homologous chromosome, which is regulating this particular phenotype, gets their inheritance and their passing on is independent of passing on of those homologous chromosomes which have the alleles at a certain locus for seed color. That is the law of independent assortment. That during gamete formation, segregating pairs of unit factors assort independently. What it means to say is that, for example, if these two alleles which are present at one particular genetic locus, right, two alleles present at one particular locus of a homologous chromosome that regulate one particular trait, for example, height, is not influencing the inheritance of a different pair of alleles in a pair of homologous chromosome that is regulating a different trait, for example, seed color. So seed color inheritance and height inheritance and any different phenotype inheritance is independent of one another. So that is independent assortment. So this law of segregation and law of independent assortment are kind of the two most important postulates of Mendel, along with these observations that that these organisms, that at least a pea plant, which this also you have to always keep in mind that all of Mendel's postulates and observations are valid, also given the background that he studied the pea plant, right? So the pea plant is a diploid organism. For each chromosome, it has two copies, right? That means at a single locus, it has two copies of genes. And these units or factors does occur in pairs. If there are two unidentical or non-identical factors present at the particular locus of this pair of homologous chromosomes, then one will be dominant, other will be recessive in terms of whether it is getting expressed in terms of the phenotype of the organism. Yes, so basically, I have already mentioned this, like kind of explained this slide. So the independent assortment is possible in this manner that, for example, here in this example, for example, you can see that in chromosome 1, you have the flower color governing genes, right, in chromosome 1. Whereas, for example, you have the pod color governing genes in chromosome 5. That's why this, like the inheritance of flower color genes and the pod color genes are actually independent of one another. Now, Mendel, to be honest, in this regard, was lucky in some sense to observe the independent assortment of the genes that regulate these particular phenotypes. Because in some cases, in reality, what happens is that some of the genes that regulate different phenotypes may be actually located very close to one another. Okay, like two genes, for example, the seed shape in this case is different. In case of P, because of Mendel was different than pod color. But if you imagine if the seed shape governing genes were very close to this pod color governing genes, then there is something which is called linkage. That means that that the recombination doesn't happen between these genes which are placed so close to one another. So mostly what happens is this entire that these genes move together. That means the seed color, one particular seed color and one particular pod color genes are not segregated independently, but segregated together. So, so that is a different case. That actually becomes a part of the non-Mendelian genetics, where independent assortment is not seen to a certain extent, and there comes a concept of linkage, which also was a remarkable discovery by Thomas Hunt Morgan in Drosophila melanogaster. Okay, for now, this is, I mean, if you try to discuss Mendelian genetics, this is an indispensable experiment. It's a monohybrid cross, right? So now you also know that Mendel used cross fertilization always. So what he did, so initially he had these pure parental lines. These pure parental lines were formed as a result of repeated self-pollination. Okay, these pure lines were obtained because of self-pollination, but then these monohybrid crosses are all cross-pollination. So initially he had this pure parental generation where he has a tall plant which has two. So basically, like there will be a pair of homologous chromosome, and this is the locus which is the genes that encode for the plant height, and this is the tall plant. So it's a pure generation, pure parental generation. So identical pair of alleles which are capital T, capital T. It means both the genes are encoding for the tallness of the plant. Whereas in this plant, you have again a pair of homologous chromosomes, but there, so again identical pair of alleles, but this small t, this is the recessive one. But small t actually means that this particular gene variant has one mutation which is not able to encode for that particular protein or that particular gene product that is necessary for the tallness of the plant. So since that gene product is not there, that's why this plant is actually not tall. Okay, but since both these copies, right? So basically, since both the copies of the gene, that is both the alleles, are this recessive, that is the mutant one. So there is no gene product, no formation of the gene product that is necessary for the tallness of the plant, and that's why the plant is small, right? I mean dwarf. So when this tall plant is actually crossed with this, is cross fertilized with this dwarf plant. So in the gametes, you can like in the first case, you can very well understand the gametes will have here all the gametes will have capital T, right? Here all the gametes will have small t. Now this is for the first filial generation, all the plants had capital T, small t. Now this is the genotype, but what Mendel observed is that phenotypically all the plants in the first filial generation were tall. So as if this recessive dwarfness is lost in the first filial generation. That Mendel realized. Now, if you see the gamete, like what are the possible gametes that can be formed from the first filial generation plants, then like the gametes can have capital T or the gametes can have small t, right? Which is also shown here in the Punnett square. The gametes can have capital T or small t. Here also it can have capital T, male gametes and female gametes, right? Capital T small t, capital T small t. So that will lead you to this particular combination of genotype that is possible in the second filial generation plants, in the F2 generation, right? Now, so actually in this first filial generation, why we are having only tall plants is because this, so in in a pair of homologous chromosome at this particular locus, which is necessary, which is regulating your plant stem height, you have one capital T which is encoding for the gene product. Okay, which is here you see there are unidentical alleles. There are two different alleles, but one is getting expressed in terms of the phenotype, the other is not. So that means this one is the dominant one, and this is the recessive one, and that is happening in terms of in the actual molecular biology picture, because the capital T gene is capable of encoding for that gene product, be it an RNA or protein, that is needed for the stem growth, for the stem, that is, and because of that the stem height is longer and the plant is tall. Now since in this case it has at least one functional copy of the gene, that is one gene allele which is functional, here the plant is tall, right? Whereas in case of recessive, in this recessive homozygous plant, since no copy was no functional copy was there, it was dwarf. In the F2 generation, what happens is again Mendel gets back this recessive dwarf plant. So because of that, Mendel could comment that this recessive gene thus is not lost. Like in the first filial generation, it's not there. It seems like the gene is somehow destroyed. The gene is somehow lost. That variant of the gene or the allele is somehow lost. But when he gets back this recessive dwarf plant again in the F2 generation, he was convinced that this law of segregation basically, that these genes do not mix or contaminate with one another. They always retain their individual identities. Okay, and they're segregated randomly and individually into the haploid gametes, right? And there are some important numbers. There's a phenotypic ratio in the F2 generation is 3:1. You see 3 tall plants and 1 dwarf plant. But if you see the genotype, then one plant is capital T, capital T. 2 of them are capital T, small t. That is heterozygous, right? So one is heterozygous tall. One is one is homozygous tall, capital T, capital T. One is homozygous recessive, dwarf, right? And 2 are heterozygous tall. That means in a pair of homologous chromosome, at the locus that regulates the height of the plant, you have one capital T and one small t. So the genotypic ratio stands as 1:2:1 for capital T, capital T, capital T, small t, small t, small t. This is a genotypic ratio for the monohybrid cross. Okay, so any questions on the monohybrid cross? You could understand? Okay then. So moving on. So these are kind of the summary of the points that Mendel mentioned, and later on, what from like Darwin's observations, all helped us understand this thing that like there are indeed different mutants or variants of one particular gene that is responsible, that regulates one particular phenotype, and only because these variants exist, like natural selection can function. How does natural selection work? It works by this manner that since there are different variants or different mutants of a one particular gene, so these individuals have different phenotypes. Like the genes or the genotype actually gives us a certain phenotype. If the phenotype is useful in adapting or adjusting to one particular environment, like one particular natural condition, then the organism is better able to survive and get access to resources and reproduce and produce more organisms, more offsprings, right? That's how that particular organism is selected. Whereas if one particular mutant or one particular variant is not giving any advantage, then or providing you some disadvantage in adapting or adjusting to one particular environmental condition, then you are not naturally selected and you are wiped out of selection. Basically, you die. The organisms die. They're not able to reproduce and give rise to more organisms that have those genes or mutants. I mean, those gene alleles, basically. And eventually, these observations of Mendel and observations of Darwin led to the this modern understanding of biology, which is called the modern synthesis. Now, just like I mentioned, that there are a large number of cases. So Mendel's rules are based on a particular system, which is the pea plant, and even within the pea plant, he examined only a certain set of heritable traits or phenotypes, and he postulated these patterns of inheritance, right? But there are also several other aspects where Mendel's trains of inheritance do not hold valid because of important differences in the molecular biology of those processes. So those are the deviation, are kind of genetic events which Mendelian genetics cannot explain, and are called the non-Mendelian genetics, which include incomplete dominance, right? Like we have all in Mendel's postulates, we have seen dominance and recessiveness. It can be dominant allele or it can be recessive allele. But in there are these non-Mendelian genetic cases of incomplete dominance, right? Or co-dominance. That means one particular gene is not turning out to be completely dominant on the other gene for some interesting reasons. And also a kind of deviation from the independent assortment law is the linkage. So which Thomas Hunt Morgan discovered in Drosophila melanogaster. So this linkage is one phenomena where, for example, on one particular chromosome, there are several different loci. That means several different genes which are clustered very closely to one another. So it often happens that often is found that there is not a considerable amount of chop and change between these genes, and these genes kind of move, like as groups or linked genes. They're called linked genes. So that the independent assortment is not seen. So their assortments are often like in terms of clusters or have a bias. So that is also a variation from Mendel's law of independent assortment, and is an example of non-Mendelian genetics. Okay. So one more interesting point where which is kind of on contrary or in contrary to Mendelian genetics. That is, there is something like there is something called horizontal genetics or horizontal gene transfer. So there is vertical gene transfer and there is horizontal gene transfer. So what does vertical gene transfer essentially mean is typically what we have already, we have been discussing, that you have a mother organism, I mean, you have the parental organisms, you have the uh, father, you have the mother, and from them the genetic information is being passed on to their offspring, right? The child, I mean, their offspring. So that is a vertical transfer of genetic information or vertical genetics. But in several organisms, especially lower organisms like prokaryotic organisms, like in case of bacteria, there is this horizontal gene transfer. So in horizontal gene transfer, what happens is you must have heard about these things like conjugation. So there is bacterial conjugation where one bacteria can donate its plasmid DNA to another bacteria by formation of a pili. So that is direct. So these two bacteria are not offsprings, right? So there is this transfer of extra chromosomal DNA from one bacteria to another bacteria, where these two bacteria are not offsprings. So there is transfer of genetic information not only among colleagues, for example, right? Among colleagues or co-workers. So that is horizontal gene transfer, which is also seen in cases of prokaryotic organisms. So that is also something which is quite different and in contrast to the vertical gene transfer and vertical genetics with which Mendel mostly worked. Now, this is the last slide of today's discussion. So this may help you understand in some like examples or events of non-Mendelian genetics, especially these events or examples like incomplete dominance and codominance. So what happens is, see, this dominant and recessive, these terminologies are relative, and in terms of molecular biology, what they essentially mean is that so these dominant and recessive terminologies have come from this fact that out of two different alleles of one particular gene, if one allele is able to regulate or get expressed in terms of phenotype, then it's called the dominant. Whereas if it is not, it's called the recessive. So now let's look at why one particular gene is dominant and one particular allele is dominant and one particular allele is recessive. So basically, alleles are variants of the same particular gene. So what are variants? Variants mean mutations, right? Changes in the nucleotide sequence. That is the only thing that is giving rise to variants. So the wild type allele here, the wild type allele is the allele which encodes for one particular gene product, right? Here, let's say the gene product is one particular protein, which, for example, is one enzyme that catalyzes one particular chemical reaction and therefore leads to a certain phenotype, because that biochemical reaction is necessary for giving rise to a certain phenotype. So wild type allele, right? Wild type allele gives rise to a certain functional protein which catalyzes one particular biochemical reaction, and that's why there is this wild type phenotype. Now what happens is there can be mutations to the wild type allele. Okay. Now, if there is such a mutation, okay, there can be, first understand the most drastic changes. Okay. So, for example, you think of this scenario that there is one particular mutation. Okay, not this scenario. This though we discussed. You consider this scenario. Okay. So there is one particular mutation which is a severe loss of function mutation. So, or amorphic mutation, because of which what will happen is that the protein that is generated is completely non-functional. If it is completely non-functional, then one particular biochemical reaction cannot at all be catalyzed, and therefore there is a loss. There is a complete loss of phenotype, or in other words, there is a severe mutant phenotype. So, for example, this wild type phenotype is your tall plant. Okay, and this severe mutant phenotype which is basically loss of function. Like loss of tallness is basically dwarfness. So this is your dwarf plant. So in dwarf plant, what we are seeing, the genotype has to be small t. Only when both the copies of one particular gene, that is both the alleles at one particular locus of the homologous chromosome has suffered this particular mutation, then none of these alleles is able to encode for that particular protein, which is necessary for the biochemical reaction that is necessary for tallness. So there you are seeing this dwarf plant, right? Which is this example that both the genes have suffered this severe mutation, because of which you have the complete loss of one particular phenotype. But again, in this plant, just think like experimentally, if you are able to introduce this wild type gene, then this gene is not able to encode for the functional protein, but this gene will be able to encode for the functional protein, which will be able to drive the biochemical reaction and therefore cause a particular phenotype. This is the classical example of capital T small t. You remember? So there, this is not able to encode for the gene, but the capital T gene is the wild type gene. So it is able to encode for the normal product, which is catalyzing the biochemical reaction and therefore causing the tallness of the plant or tall plants. So that's why these plants were tall. And you can also now understand that why capital T is dominant, because this is a wild type gene which can encode for a functional protein or functional enzyme for that case, for example, which can catalyze one particular biochemical reaction and therefore drive one particular phenotype. That's why this gene is not able to govern the phenotype, but capital T is, right? So these are the most classical scenarios. Now you get to see some like, and biology is never black and white, right? It's always gray. So you get to see intermediate steps. For example, there is one particular mutation. Let's say in this case, there is one particular mutation where you have a less functional protein. What do you mean by less functional protein? For example, it's an enzyme that is still able to catalyze a particular chemical reaction, but not with that high efficiency that the wild type protein does. That is, it has a lower efficiency of catalysis, right? So because of that, you may have a mild mutant phenotype, right? A mild mutant, when will you have? When you have this homozygous, right? Homozygous mutation, like homozygous, like you have both the alleles which has this mild mutant gene. Again, if you have one particular wild type gene, it will be almost overcome. Or if there is a very high, now there is also one concept called dosage dependence, that if if the concentration of the enzyme that is necessary for driving the biochemical reaction is very high, then even if you have this one particular wild type gene, it might not be sufficient to compensate for this particular mutant. So you may still have some recovery, but still not the same phenotype as the wild type. Okay, so there comes a grayness in biology. And there can be one scenario which is very interesting, which is called the dominant negative. So this is a very interesting scenario. Here what happens is there is some particular mutation in the gene, which is now encoding for one particular protein, which is inhibiting or interfering the function of the wild type product, right? So this is your mutant product, this one, which is completely inhibiting the activity of the wild type protein. So here what will happen, that even if you have this like capital T small t scenario, here this small t is this dominant negative. If the small t is dominant negative, then what it means is that it is encoding for one particular product which is completely inhibiting the normal wild type gene product that is being encoded by capital T. Even in this scenario, you will get the mutant phenotype. So if it's homozygous, you definitely will get the mutant phenotype. But even if it's heterozygous, you will get the mutant phenotype. Okay. And so So only if you have this wild type, like wild type homozygous, you get the normal organisms. So in case of heterozygous also, in this scenario, if you have this dominant negative allele, then it will be a problem. We will have the mutant phenotypes, right? Okay, so this is kind of a bit of molecular biology picture of what causes like dominance and recessiveness, like why some genes are dominant, not genes, why some alleles or the mutants or variants of one particular gene are dominant, some are recessive, and some are dominant, not only like, like positively dominant, some can also be dominant negative. Like they are encoding for one particular gene product, which is inhibiting the activity of the wild type. So even in heterozygous scenario, you are getting the mutant phenotype, but it's, yeah. Okay, so this is all. This is what I had from my side. If you have any questions, you can ask. We can discuss. Okay, then if you don't have any questions, then let's conclude this session for today. All the best for the assignments of this week. Um, uh,
Thank you, sir.
Again, we will. Okay, thank you. Welcome. We will meet in the next session. Okay.
Okay, sir.
Okay. Bye.
Bye, sir.